Hey all, this is Professor Curt here, lecturing on Biology 251 and Anatomy and Physiology. We're on Chapter 17, Special Senses, and I wanted to do a supplementary video on G protein coupled receptors. This is something I have a background in, and I think it really ties well into the nervous system. Um, you may or may not have covered in Chapter 9 if you took Biology 141. Chapter 9 talks about cell signaling mechanisms, and it might have talked about this. Also, um, if you go on to take AMP2, Biology 252, and discuss Chapter 18, depending on the level of detail that your instructor covers, you may or may not talk about G protein coupled receptors. So, why is this important, and why am I bringing it up in the special senses? Um, I hope to answer that in a second. So, first of all, we need to talk about, what is a receptor exactly. So, a receptor is often a protein um, that, binds to a molecule called a ligand, and then leads to a change in a cell, right? That's a pretty general definition. You have all kind of different receptors. You've got nuclear receptors located in the nucleus. You've got membrane-bound receptors. You can divide those membrane-bound receptors up into G protein coupled receptors or tyrosine kinase receptors, or just a whole multitude of receptors. You've got literally hundreds, if not thousands, of these things in your body. So why are we talking? What does it mean if a receptor is you know coupled to a G protein? What is a G protein? Why is this important in the nervous system? Well, a G protein. So we've kind of defined what a receptor is. That's a protein that binds a ligand. I guess what is a ligand? A ligand is just a molecule uh, that binds receptor. What is a G protein? A G protein is a protein. You got like some protein here. It's got a little pocket in there, and this pocket can be filled with a molecule called GDP, in which case it is inactive. And there are these other proteins called, let's see, guanylyl exchange. Uh, Hold up. Sorry about that, y'all. I blanked on the word and I did not want to have to restart the video. But they are guanine, sorry, not guanylyl, but guanine exchange factors. That's where I was going with them. Um, and these guanine exchange factors, what they do is they swap the GDP out, right? So the GDP leaves this binding site, and then it brings in a guanine triphosphate. So GDP for a GTP, and now this protein is considered active or on. And there's an enzyme typically in the G protein that will, over time, you can imagine this as um, guanine with 3 phosphates. And over time, an enzyme will cut off one of these phosphates, changing GTP back into GDP, turning it off again, right? So now it's off. And then, you need a new guanine exchange factor to swap out the GDP for GTP and turn it on again. And typically this thing is going to bind something downstream and do something. All right, so that's what a G protein, that's what a ligand is, that's what a receptor is. G protein coupled receptors basically are a link between the ligand and this G protein. Basically, it allows the ligand to turn on this G protein coupled receptor, and the G protein coupled receptor basically acts as a guanine exchange factor, turning on your G protein. But it only does that when the ligand's present. It'll make more sense, I promise. Why are G protein coupled receptors important? Well, it turns out if you go back and read in the past, you might have read about metabo. Like if you read chapter 12 very carefully, you might have read something about metabotropic um, uh, ligand gated ion channels. These are typically G protein coupled receptors, right? Meaning that G protein coupled receptors are often are involved in many neurotransmitter receptors. Also involved in the special senses, right? Like opsins that we'll talk about in a bit. They're involved in, opsins are involved in vision. So your receptors for vision are G protein coupled receptors. Your approximately 3% of your genome, you have about 1,000 G protein coupled receptors involved in olfaction, right? They're also involved in most of your sensory modalities for gustation. Hormones, right? Many hormones, including insulin, melatonin, come to mind. Many hormones act on a G protein coupled receptor pathway. And at one point, and this may be outdated data, but at one point, 70% of drugs in development in some way or another acted on G protein coupled receptors, right? So they're important. And since we're talking about the special senses, you know, opsins are G protein coupled receptor involved in vision. Your olfactory receptors rely on G protein coupled receptors, and same thing is gustation. And rather than explain the pathway every time we get to olfaction, gustation, and etc., making this whole separate video. So, how does a G protein coupled receptor work? Well, kind of depends. There's several different, I guess, pathways. There's several different types of G proteins they can couple to. If you want to read more about this, I'm going to be talking specifically about G alpha S. There's also G alpha I and G alpha Q. But basically, this type of receptor is found in the membrane. So we can represent like a phospholipid bilayer, and embedded in it, we have a G protein coupled receptor. So I'll pick a different color for that. G protein coupled receptors, if you go back to this picture here, you can count them. There are 7 alpha helices that cross through the membrane. So sometimes these are called 7 transmembrane proteins. We often represent them as a little squiggle like this because this has 1, 2, 3, 4, 5, 6, 7 transmembrane domains, and we may draw something here to represent where our ligand binds, and this tail here might represent the part that um, interacts with a G protein. So that's what we'll draw, right? We've got our 7 transmembrane domain. We've got a binding site for our ligand. Well, I guess we'll use a different color maybe for the ligand. And this thing is going to be coupled with a G protein, so maybe we'll use a different color. These G proteins have a little hydrophobic tail made of a lipid that allows it to embed within the membrane. And then, of course, it has a little binding site for some, GDP or GTP. We'll use we'll start off with GDP in there. Maybe I can write that a little more clear in a different color. Here we go. We're going to start off in the off state, all right? So it's got GTP. And basically what's going to happen is our ligand if the ligand is at low concentration, or if it's not there, then it's not really going to bind the receptor. But as the concentration increases, at some point there's going to be enough of it around to start significantly binding up receptors. Or you got to imagine there's like hundreds or thousands of these receptors in the membrane, or at least dozens. So we're going to imagine, okay, there's enough of this ligand there to bind the receptor, right? What that does is it causes a change in shape to the receptor, such that the receptor acts as one of those guanine exchange factors, and our GDP is going to leave our G protein. And now let's use a different color. Maybe, I don't know, maybe this bright red. Some GTP now fills up our G protein, right? So now our G protein is on. Now, once this G protein is on, it actually is able to leave the receptor and diffuse around the membrane. So it actually like moves through the membrane, and it's a little bit more complex than this. There's actually 3 subunits, and they can break off, and different subunits can do different things. But we're trying to keep it simple here. So now we've got our G protein is on. Oops, wrong button. And it's diffusing, and it's going to bind up an enzyme in the membrane, right? So in the membrane, there is some enzyme. Now, in the case of G alpha S, if that's what this G protein is, often this enzyme is an enzyme called adenylyl cyclase, right? So adenylyl cyclase, this is an enzyme, and as you may remember, enzymes carry out chemical reactions. So there's kind of an interaction here between this G protein that's on and this enzyme. Basically, the enzyme is going to become active, and it's going to start converting the cell's ATP into something called cyclic AMP, right? So just quick recap of what ATP is. You might have learned this in biology. It's kind of this nucleotide triphosphate that the cell uses for energy. So there's always lots of it around. It looks something like this, with not 1, not 2, but 3 phosphates, right? It's tri, meaning 3 triphosphates. And there are these hydroxyl groups down here on the ribose, right? Basically, what this enzyme adenylyl cyclase does is it breaks this covalent bond here and forms a new covalent bond between that phosphate and this oxygen here. So now there's this cyclic part of the molecule, and since there's only one phosphate, we say that it is a monophosphate. So hence cyclic adenosine monophosphate. That's where we get the name from. Now, this cyclic AMP, right? The concentration is going to increase very rapidly, because you're turning, this enzyme is going to convert hundreds, if not thousands, if not hundreds of thousands of molecules very quickly. And this cyclic AMP, as it builds concentration in the cell, it can do all kinds of different things. There are transcription factors that can turn on and change cell signaling. There are enzymes that can get phosphorylated and turn on, like in a liver cell. Like when insulin goes through this process, it'll turn on the enzymes that make glycogen, right? But we're talking about neurons, and the main thing that this cyclic AMP is going to do in a neuron is it's going to bind an ion channel. And when the cyclic AMP binds that ion channel, the gate that was keeping this ion channel closed is going to open. All right, and like we saw in chapter 12, once ion channels open up, ions, typically maybe positive ions, may come into the cell and lead to a depolarization. Right. So basically, what we've done is we've linked a very small amount of some ligand to the depolarization of the cell. The depolarization, of course, is going to lead to an action potential, and the action potential is going to lead to a release of neurotransmitter. So this is going to become very important in the special senses because two of them are chemical senses where you're directly detecting a ligand. Like this ligand could be, for an example, fructose. Right? Fructose is a very sweet sugar, and this fructose could bind the receptor very tightly, depolarize the cell, and you would send a signal to your brain, and you'd be like, okay, I'm tasting something really sweet. Or this ligand, as we're going to see in vision, could be a molecule that light destroys. So you have a way to detect if there's lots of light, because in darkness, you're going to have lots of this ligand around, and this is going to tell your brain, I'm seeing darkness. And then as the light destroys the ligand, it's going to shut this whole signaling pathway down. And it's basically going to tell your brain, I'm seeing lots of light. So we'll see variations on this pathway several times. I included this slide. I don't know how well you can see it because my webcam's covering up a little bit of it. But this right here, this I included this image because this is what the protein in the membrane actually look like. Like these are molecular images. So this is our hormone, which is acting as our ligand, right? This is the G protein coupled receptor, right? This is the G protein. And all of this is the adenylate or adenylate, depending on who you ask, cyclase. And so they've got the steps here where the hormone's binding the receptor. And, here we can see the GDP exchanging for the GDP, and it looks to be the alpha subunit. This is the beta and gamma subunit here and here. The alpha subunit is activating the enzyme, which is converting ATP into cyclic AMP. So anyway, wanted to do that, supplementary video in case you're interested. This type of stuff I studied in graduate school, cell signaling and hormone action. And I'll be able to reference this video when we talk about this in vision and olfaction. Maybe decrease the length of those videos a bit.